Good afternoon, and welcome to our weekly Research in Action series brought to you by the Division of Research at Florida Atlantic University. My name is Jeannie Viviani. I'm the Director of Research Development, and I will be today's host and moderator. It is my great pleasure to introduce today's speaker. Dr. Osama Refai is a neuroscientist interested in understanding how the human brain works in health and diseases. He uses multiple pharmacological and genetic approaches to uncover the genetic basis of psychiatric and neurological disorders such as Parkinson's disease, ADHD, bipolar disorder, and addiction. Dr. Refai is currently a research assistant professor of biomedical sciences at Florida Atlantic University College of Medicine and at the FAU Stiles-Nicholson Brain Institute. During his postdoctoral training, Dr. Refai worked with world-renowned neuroscientist Dr. Randy Blakely at Vanderbilt University and currently now at the FAU Stiles-Nicholson Brain Institute to identify novel regulators of dopamine signaling. Dr. Refai has a Ph.D. from the University of Calgary in biochemistry and molecular biology, a master's degree from New York University in human genetics, and a master's degree from Cairo University in physical anthropology. His research is published in well-established and prestigious scientific journal, and he serves as a peer reviewer for several scientific publications in the field. He's a member of the Genetic Society of America, Society for Neuroscience, and the American Association for the Advancement of Science. Dr. Refai, welcome, and the floor is yours.
Thank you so much, Jeannie. I appreciate the opportunity to talk to people at our community about the work that we do in the Brain Institute and the Blakely Lab. So today, I'll have the pleasure to talk to you about how we use a simple model organism, C. elegans. It's a tiny little worm that's a microscopic organism, which we are able to use to solve complicated problems in the brain. And I'm going to talk to you today, give you a brief introduction about our work, and then we'll take you through that talk with this outline. Just to start with the neurotransmitters again, here looking at a zoom-in picture for the cells in the brain, where these cells has to talk to each other using a chemical substance we call neurotransmitters. And these chemical substance, as I just said, without having the slide showed, that if you've seen the movie Inside Out, there are a lot of factors play in our brain to regulate our behavior. And these behaviors include pleasure, fear, disgust, whatever it is. And I'm interested in studying one of these molecules called dopamine, which mainly play a major role in controlling our motivation, reward, attention, pleasure, and so on. And as you would imagine, if something goes wrong with this molecule, the level of the molecules goes up, goes down. The neurons which are responsible for production of dopamine are called dopaminergic neurons. When these neurons get sick, a lot of problems happen, and these include, but not limited to, depression, Parkinson's disease, and addiction. I'm really interested in studying Parkinson's disease, and that's what I'm going to talk to you about today. So as you know, Parkinson's disease is a major issue. It's a complicated disease which start in the brain, but also have a lot of symptoms involving movement, memory, learning, and a lot of people have been diagnosed with it, including celebrities, and some of them are actively working now to fight the disease, like Michael J. Fox with his foundation to fight Parkinson's disease. So Parkinson's disease, as you know, is something that's involving a brain disorder when the brain is not able to produce enough dopamine, especially that area of the brain. So this is an MRI, very classic MRI for people with Parkinson's disease, looking at the left here where the human is healthy, and then the Parkinson's disease start to show up. And then when it advances, what you see the difference here is these red dots, which indicate the level of dopamine in the brain. So as you can see, the less the dopamine is, the more severe the disease is. So Parkinson's disease is actually expected to increase in the next few years. Sadly, the number is expected to double from about 6 million in 2015 to about 13 million in 2040. And it's the Parkinson's Foundation reported that every 9 minutes in the US, there is a new case diagnosed with Parkinson's disease. The number can reach 60,000 cases in a year. And another fact about Parkinson's disease is that this disease is an age-associated disease. So usually our senior citizen, and when people reach the age of 60 and around that, this is where the disease starts. But the factors that contribute to that could have started earlier and just gone unnoticed for so long. So the disease, the main symptom of this disease from a research point is that the disease involves death of dopaminergic neurons, those neurons which are necessary for production of the neurotransmitter molecule dopamine, which I told you about, controlling our memory, our pleasure, our movement. And when this dopaminergic neurons, as you can see, so here again, this is MRI showing that a healthy brain would have enough amount of the red dopamine here, as you can see, it's stained in red. And the Parkinson's disease patient would have less amount of dopamine. What happens is when the dopaminergic neurons are not able to produce enough dopamine, like in normal conditions, they are not able to send the right message to different muscles and different motor neurons. And that's what causes loss of control on motor activity. But also that might start causing neurodegeneration, where the neurons themselves start to die because they are not able to sustain their ability to secrete dopamine and to perform normal function. So that takes us to just trying to look at this problem from different angles, trying to divide and conquer this very complex disease. And if you look at our brain, the human brain is a very complicated machine. It contains millions of neurons. As you can see here, these are millions of neurons with their axons and millions of circuits and connections, which is very, very complicated to study. Parkinson's disease involves only one subset of these neurons, which are the dopamine neurons. So to make this problem easy, I usually ask my student this question: if you want to understand how an engine works or how a motor works, would you start with an aeroplane engine that's very complicated, very sophisticated, or would you start with a simple model for an engine that only have few circuits and tiny wires, and that would be easy to dissolve, easy to reassemble, and easy to understand how to work? Most of my students, smartly enough, they pick the simple model to start understanding how to work first. And this is what we do in trying to understand how our brain works. So by looking at our brain here again, if you zoom in, the more you zoom in, you will find a lot of networks, a lot of complexity. The number of dopamine neurons themselves is about 500,000 in our human brain. And that led us to find a very simple model, which has only 8 dopamine neurons in their brain. And this is the worm C. elegans. So this is a microscopic worm. As I said earlier, it's just about a couple millimeters you see under the microscope. And using GFP fluorescent protein technology, you are able to visualize these neurons under the microscope while the animal is living, meaning that we can study what happens to these neurons, how they develop, what happens if they got disease, how they die. And being very simple, being able to look at every single neuron individually is an advantage that you do not have in human or other organisms like mouse. That model organism, C. elegans, provided 6 people, there are 6 scientists so far, have won Nobel Prize for big discoveries in the field of medicine and physiology for the programmed cell death, how cells die and how cells are programmed to die, how cells start and develop. Also another known, now widely known phenomena, RNAi and the green fluorescent protein, which I just shown you, the green neurons. And there is more to come, I promise. So we just talked about how simple is the brain of a worm that would allow us to look into the dopaminergic neurons in more details and study them. But not only that, there is another advantage here where the chemical molecule, the neurotransmitter dopamine, is the same chemical molecule as it is in the human brain. So you would imagine that it would have the same function. And I'm going to talk to you about this a little bit in a little bit later. More to that, the genes which control all dopamine related process, from dopamine synthesis to dopamine secretion to dopamine clearance from the system, these genes are highly conserved to the human genes, which means that they have huge similarity, meaning also that when we study one of these genes in a worm, we'll be able to apply the same to human genes. Another advantage, which comes especially when you study diseases like Parkinson's or Alzheimer, that these animals have a very short life cycle. Parkinson's disease in human, as we said earlier, only appears at old age, starting 60 and later. And to wait for a human to age to be able to study that the dopamine neurons or Parkinson's disease, that's going to take years or decades, and it's impractical. So that's why scientists use models like C. elegans. In C. elegans days, the whole animal or the whole life cycle of the animal can span about only 2 weeks. So in 2 weeks, the animal starts as a child and then reach maturity, adulthood, and then reach aging stage in just a few days. And that would allow us to track neurons and see what changes happens to them in a such faster way than what would be the case in human samples. Adding to that we were able to mimic Parkinson's disease in these animals. So the way we do that now is one of the ways, actually, there are multiple ways now that we can use to mimic Parkinson's disease in these worms. But one of them is that we were able to use neurotoxins to kill dopamine neurons and then see how the animals behave, how does it react, what genes are involved in this process. And this allows us to understand what happens in Parkinson's disease. So using this chemical substance, which is 6-O-HDA, it's an oxidized dopamine, which when the animal uptake this chemical substance, we see that their dopamine neurons disappear. So what you see in the left here, you see that this is a normal case animal where they are healthy, have healthy dopamine neurons. When you treat them with this chemical 6-O-HDA, you will find that their dopamine neurons dies. And that's actually mimicking what we see in Parkinson's disease. And that was a tool that been used by us and by other groups to discover new genes that are involved in regulation of Parkinson's disease, and in particular, protection against the neurotoxins. As we said earlier, dopamine as a molecule is the same in human as it is in C. elegans. There's no difference, and that actually leads to similar functions. So in C. elegans, it does also regulate the learning of behavior of the animal, their ability to move, and one of these movement abilities is their ability to swim. And I'll tell you how we use this phenomena or this fact to study dopamine neurons in C. elegans. So number one, as you see, we are able to see dopamine neurons in C. elegans, and we will be able to tell if they die or not, if they are healthy or not. But also we are able to see if the animal is able to move and swim, meaning that they have proper dopamine signaling, or if they are not able to, that would mean that they lost their ability to regulate dopamine in their brain. And this video is showing you a real experiment from the lab where we give these animals, in the animals that are healthy and have no problem with their dopamine, when you incubate them in water, you'll see that they are swimming fine with no problem for an hour. They swim with no problem because their dopamine regulation of movement is intact and is fine. But when these animals lack an important protein in dopamine regulation called the dopamine transporter protein, they lose their ability to swim within few moments. So this is an actual time. It didn't go for more than 30 seconds now. And you see the animals lost their ability to swim. And that's what we call something can be similar to what we see in humans with problems in movement due to Parkinson's disease. So this data, we are able to quantify in graphs to publish our work, and just showing how many animals can swim versus how many animals are not swim. But also we do this using computer algorithms and softwares that allow us to measure every single thrashing and frequency of the thrashing of these animals and track their ability to swim, which is meaningful in understanding what genes are involved and how their dopamine levels are affected. And we use this phenomena to discover more genes involved in dopamine signaling. And the way we do this, we do it through a way called forward genetic screening, where we treat these animals with a drug called the EMS. So this drug is a carcinogenic substance which causes mutations. It breaks the DNA. It causes cancers in those animals randomly. And it hit randomly through the genome. It hit genes randomly. And then we take those animals and put them in a swimming test, as you've seen earlier. And those that are not able to swim, we call them SWIP, which means they SWIP-induced paralysis. So they are not able to swim due to probably dysregulation of their dopamine and inability of the dopaminergic neurons to control their swimming behavior. So we take these animals as potential candidates for novel genes that are involved in regulation of dopamine signaling and possibly involved in Parkinson's disease. Using this technology, we have uncovered so many novel mutants, and we have published on that over the last few years. And today I'm going to talk to you about one of those genes, novel genes that we have discovered through this technique. So this novel gene is called SWIP20, and SWIP, as I just said, is the inability of an animal to swim due to disruption of dopamine signaling. So looking at this animal in particular, or this mutant in particular, you will see that the normal animal are able to swim about 100% of the time with no problem. So the red here in these heat maps mean that they are able to swim throughout the time with high frequencies. The lower the frequency of swimming, the green, the light you see. So here, when these animals have this mutation, SWIP20 mutation, they are not able to swim, as you see here. And that's very similar to the animal that we shown you earlier in the video, which has a known mutation in dopamine regulation protein called DAT1, or dopamine transporter protein. You can see this here depicting the frequency of swimming of these animals. So if you look into the wild type animals in black line here, they swim very high with no problem. But the mutant animals which have mutation in their dopamine signaling, they are not able to swim or have very, very low swimming frequencies. So one of the questions that we ask ourselves when we get such mutants is this mutation, or is this paralysis we've seen in water, is due to the animal having really a problem in their dopamine signaling, or it could be a problem in their muscle? They might just have some muscle spasm, some muscle defects that are not allowing them to swim, like someone who's just running and not able to go through the marathon because she is tired and she just needs to take a rest, or it could have a heart attack, right? So these two conditions, to differentiate, we use our knowledge of pharmacology and drugs that already been prescribed and FDA approved drugs for patients with neuropsychiatric disorders, including risperidone and azaparone. So we use these drugs to treat these animals and see if these drugs were able to help the animal swimming better. That would mean that the mutation we got is actually in a gene involved in the dopamine circuit. But if the drug is not able to help the patient swim, or the mutant animal here in this case swim better, it means that it's not involved in dopamine. The mutation is not involved in dopamine signaling. So this is another drug that I've used here called azaparone, and we have published on that in 2019. So this is the regular case, when we have the SWIP20, the mutant, the novel mutant, we isolate it from the screen. As I shown you earlier, the green means they are paralyzed. They are not able to swim for all the time that we record their videos. But when we add the drug here, the azapiron, they gain their ability to swim in a matter of few seconds, meaning that they have no problem with their muscles. They have no problem with their swimming circuit in their brains, but they only have a problem with their level of dopamine. And when we give them this drug actually reverses the dopamine, the problem with dopamine levels, and allows them to swim. So this is a confirmation that the mutant we got is for a gene involved in dopamine signaling. So from that, we've asked the question, where does this mutant function? What kind of mutant it is? What kind of gene is it? And we have some science by studying the symptoms that appear in this animal, or some diagnosis of the animal's ability to swim, the ability to do normal functions, to learn to move, and so on. We've done some, chemical assays and some experiments that indicated that they have a low energy levels in general. So these animals have low ATP levels. They have a high oxidative stress, meaning that they have free radicals in their cells, which actually probably contributing to the death of dopaminergic neurons. They have low levels of oxygen, and they have few ability, or actually lower ability, to consume oxygen. And these are all symptoms of defective mitochondria, or a problem with mitochondria. So what is mitochondria? Mitochondria is that small organelle in the cell, in any living cell, actually. There's it's called the powerhouse of the cell. It's a tiny organelle there, but has major function in generating energy. So it is charger of the cell, let us put it this way in iPhone terms. So when an animal or a neuron has defect in these mitochondria, it won't be able to produce energy. It won't be able to sustain the fuel necessary for a neuron to fire, to work, to sense, and so on. And as a matter of fact, our brain is a huge energy consuming organ. It consumes more than 20% of our body energy. So you would imagine if you have a problem with mitochondria in those neurons, there will be a huge problem in our brain. And neurons in particular are known to have huge number of mitochondria comparing to other cells, because they need a lot of energy. So again, we have this scenario now that we're working with, that probably those animals and probably patients with a mutation in that same gene, they have a problem in their ability to fuel their dopamine neurons. So the neurons are not firing properly. They are not able to secrete dopamine properly, or they are not able also to clear out dopamine properly from the system. And it's a known fact that when mitochondria is dysfunctioning or is broken, neurons tend to die. And neurons of patients with so many diseases, neuropsychiatric diseases, usually have a problem with their mitochondria, including Alzheimer's, Parkinson's disease. All aging diseases usually involve a problem with mitochondria. So for us here to understand and to show for a fact whether the new gene we have discovered is causing a problem in mitochondria of the dopamine neurons, we wanted to know whether the neurons actually are dying or not. And we've done this assay. So what you're looking at here in the left at A, you're looking at the healthy neurons of a brain of a healthy worm. When they have that mutation, the novel mutation we discovered, they actually start to die. So neurons starting to die. It's shown here in this cartoon at B. So the green is a healthy looking dopamine neuron, but the black is non-healthy, a mutant dopamine neuron where they don't have that gene in their mitochondria, and the neurons start to die. And this is quantified here in CDE. As you see, the number of animals that has death of dopamine neurons are about 80%, comparing to the wild type animals, which has almost zero of dead neurons. And with our collaborators at Rutgers University, we have developed a technique to look into the mitochondria of those neurons specifically. And the way we do this is we use the GFP, the fluorescent protein technology, to light up the mitochondria of the dopaminergic neurons of these animals and to study them. We take this a step further with our collaborators at Columbia University as well to study the metabolomics of this mutation, what energy deficit that have happened, what kind of molecules that have actually been missed, and what pathways that were upregulated or downregulated. Just to make the long story short here, if you look at these images, we study here their mitochondria, and these tiny, tiny green dots are actual mitochondria of the dopamine neurons in the worm. And when you compare what you see here in the wild type or the normal animal, you'll see that their mitochondria is green, visible, and we call these healthy mitochondria. But when you look in the ones for the mutant, the gene that we have discovered when it's mutated or absent, you will see that mitochondria is represented as very, very tiny circular dots, which are actually fragmenting, disappearing, and that means that these mitochondria are sick, are not able to fuel the dopaminergic neurons. And this is just maximized here. So looking at a healthy mitochondria here in a tubular, healthy, good-looking shape, they have enough number of mitochondria. But the mutant, the gene, when the gene is lost, you'll see that there is less number of mitochondria. The mitochondria are smaller and circular in shape, and this is a sign of sick mitochondria. And another fact, I don't want you to worry too much about the details of this figure, but another thing that we have discovered, that this loss of mitochondria is happening actually based on the age. So the older the animals get, the less number of mitochondria they have in their neurons. And this is a known phenomena in a lot of neurodegenerative diseases, which are age-associated, including Parkinson's disease. So what we see here, animals at very, very young age have very healthy-looking neurons, wild type, and also the mutant have very good number of mitochondria at early ages. But when they age to about day 12, which is really, really old for these animals, is like you're talking about 70 or 80 years old for a human, you'll see that the number of dopamine neurons that have mitochondria is decreasing. You can comparing the wild type here, which is normal just for the last day here, to the mutant case. You'll see that the mutant has almost, we can barely see any mitochondria left there. So this is another sign that this disease is very similar to what happens in Parkinson's disease patients. So to take this step further, what does it mean? We found something in worm, in C. elegans. What does it mean to us humans? How can we learn from this, or how can we benefit from this? So to do this, the first thing that we do is we take the gene that we have discovered in a C. elegans and do something similar to Google search in the genome of the human. So you take the sequence of the gene, which is something similar, like a DNA sequence like this, and you search the human genome for a gene that's similar to that. And we were able to find that this gene we discovered in worm exists in all forms of life, not only humans, exists in all forms of lives that have mitochondria, and it exists in human. So this was very, very good news to us. And this is the sequence homology thing I showed you, just showing you that it exists in all form of life, from bacteria to primates or mammals, including human. So why this pathway is everywhere, why this gene is everywhere, why it's so important? We find out that it's so important because it plays a role in the production of a very important natural product called alpha-lipoic acid. And alpha-lipoic acid, some of you might have seen it in the multivitamins stores and shelves there, that some people take it as a supplemental nutrient or food supplement. I'm not prescribing this here now. This is not a clinical show, but it is been used before as an antioxidant. So it helped eating up the free radicals and oxidative stress from your neurons, and it helped people so much, but it wasn't known why it does this function. It could be so many things. But today we have found out that there is a specific gene that's necessary for production of this natural product, the alpha lipoic acid. And from that, we thought, would we be able to use this to treat our worms? And that will be promising for us also to produce something that's a strategy to help alleviate symptoms of Parkinson's disease. More studies actually came for similar genes to our gene, describing humans with intellectual disabilities, motor disorders, symptoms similar to neurodegenerative diseases and things like this. So that was a good indication that we are in the right direction. And here I want to talk about our efforts to make use of our discoveries here to help people with Parkinson's disease. So Parkinson's disease have so many symptoms. It's a very complicated disease. And one of these symptoms is the existence of a toxic protein in the dopamine neurons of the patient. So this toxic protein is called alpha-synuclein. Alpha-synuclein is a normal protein, but in the patient, it exists in massive amounts and start gathering and aggregating and forms very long fibers. Those fibers stiffens and kill the cells. So they do kill the cells of Parkinson's disease patients. So we thought to use this protein and introduce a Parkinson's disease-like worm. And that was actually done by other collaborators. So we've got this worm which has Parkinson's disease in their dopamine neurons or in their brain. So here, if you look into the healthy looking animal here, it's labeled as N2. So these have very healthy looking good dopamine neurons. But when you put the alpha-synuclein, this toxic protein on their dopaminergic neurons, you will see clearly that a huge subset of those neurons is dying, and the neurons are completely lost, as you can see here. And the more the time goes, the more the loss you see in dopaminergic neurons. So now we have a worm, the worm UA44, or this animal is a model for Parkinson's disease. Can we treat these animals with our gene that we have discovered, or with the natural product of it, the alpha lipoic acid? And that's what exactly we have done. And giving these animals so again, just walk you through this again. This is the same thing you've seen earlier again. When you give these animals the gene again in a gene therapy like technique, you provide them the gene because they have a lack of the gene, they have a mutation, their gene is not functioning, so you inject the gene into them. So when you inject the gene into them, what you see is they have less number of dead neurons comparing to the ones that are sick and do not have the new overly expressed gene, or the gene we introduced into them. But also when you give them the alpha lipoic acid, just as a simple food supplement. So we incubate them for long enough time with this drug, they eat it, they absorb it, and then we count their dopamine neurons that are healthy. What you have seen that there is a significant decrease in the number of dead neurons. So we'll see more neurons starting to survive and to fight the alpha-synuclein, this toxic protein, which actually give us an avenue to move forward and propose that this can be done in human, but that would would need a lot of funding. It would need a lot of trials to show that this is that would be helpful for people with Parkinson's disease. Again, I'd like to remind you that the disease is very complicated. It has so many symptoms. There is not one single gene that controls it. There is a huge network of genes. It's a multifactorial disease. But we think a discovery of that gene or a gene like that would bring us a step closer to develop a technique to study Parkinson's disease and to help people who suffer from such a disease. So just to summarize what I have said, then we have a newly discovered gene, SWIP20, which plays a major role in the production of a very well-known natural product called alpha lipoic acid. And these two can be used as potential therapeutic in potential therapeutics for Parkinson's disease. We've shown that we can using a tiny little worm, we can generate a model for Parkinson's disease that lead us to discover genes that most likely plays a major role in controlling dopamine neurons and in protecting dopamine neurons against neurotoxins, against factors that kill them, and also would alleviate and ease the symptoms of Parkinson's disease when you put the gene back into these sick animals, or when you give them treatments of alpha-lipoic acid. So with that, I would like to thank my mentor, Dr. Andy Blakely, for all the support he's given me over the years, and members of his group for their support and very, very insightful discussions. And would like also to thank our collaborators in Rutgers, Columbia, and FAU Scripps. And thank you to the organizers of this event, and thank you for being here and for being interested in to listen into to our work. And I'll be happy to answer any question.
Thank you, Dr. Refai. That was fascinating to understand, I guess, all the way back to the dopamine neurotransmitters. I'm monitoring now for questions and answers, but I did want to kind of address some of the things that you were talking about in terms of the therapeutics, right? And obviously people might listen to this and they latch on to things like that. Could you give me some insight from a researcher's perspective, how you go from your initial like scientific question, if you will, you're looking at neurotransmitters, and those are neurons that are not creating the dopamine, correct? And so you kind of go downstream to look at, well, why is that? And what is the mitochondrial aspect? And then you look at the therapeutics. How long have you been doing just this area of research, just by giving us that kind of thing? And then from that point, how long does it actually take to do things like therapeutics for these disease processes?
That's a really great question. Thank you. So time-wise, this project we started this project at Vanderbilt University, and when we moved in 2016 to FAU, we moved the project here, and the project is fully mature. We are in the process of publishing our results. But the time varies from a project to projects like these. It depends on so many factors that part of it is funding. Sometimes you have funding available to do, to get some reagents that will allow you to get quick results. Other part of it is just trying to connect the dots together. So this is, you looking sometimes, you dig in the desert for something. When we've been doing this genetic screen, as I said earlier, we give the animals this drug, which is a carcinogenic substance. It kills some genes, and we don't know what genes we pick in. When we got this SWIP20, we didn't know what it is. It could be something that's already known, and you just, that's it. And you could be lucky and get something that's not known before, and nobody studied it in that sense before, like this gene we've got. So it's a lot of contributing factors, but our plan was that we have had validations at every step, as I shown earlier, using antipsychotic drugs, which are already known. These were giving us hints that we are moving to the right directions. If we didn't get positive results from using something like azapiron, this is an antipsychotic drug, we give it to the animals. The animals are not able to respond. Their health is not improving. Their dopamine neurons is still dying. So if you do these things and we don't see results, we say, oh, this is not it. We're not looking for this. So we go to another one. And luckily enough, when we do these forward genetic screening, it generates to us a big library of potential novel genes. And we go through them one by one. So as a matter of fact, this wasn't the first gene I picked from this screen. I've worked with 5 other ones, and this one was number 5 in the line because the others were like false positive and they weren't giving the indicative data that we were looking for.
I see. And along the lines of gene therapy, a question came in just now. How do you expect that your prospective gene therapy may compare in efficacy to other Parkinson's disease gene therapies currently in clinical trials?
It's too early for us to compare with other gene therapy techniques in clinical trials because we're not there yet. there are some validation steps we have to take before taking this to a pharmaceutical company and say, hey, we have this product now that we think is going to be helpful. So scientifically, we need to do some few more experiments, not too long. We're right there. And as I said, this disease is a very complicated disease. It's been said that everyone or every patient has his own Parkinson's disease. It's an individual disease. And because it's a huge network of genes that are contributing to it, multifactorial. So if you have 100 patients, you might not find that they are identical in their symptoms, in their cause of disease. So ours is gonna add to the existing library of treatments and is gonna definitely help a subset of patients, not all patients. And I don't think there's a drug that exists that does that. So just we trying to put, pieces together right now.
I see, definitely. Another question came in from an Alberto Hassis. Is the lipoic acid deficiency or the dopamine production the problem with the dying cell? It looks like it is just the LA. If you provide dopamine to the neurons, do they survive? Also, do fluoroquinolones appear to reduce mitochondria damage? Can you use your system to fish out the mitochondrial damage? That's a kind of a multi-part question there. So yeah. I don't know if you need me to repeat any of it.
Let me try to answer it. And if I missed something, let me know. Okay. So the first part is, yes, when we treat animals with Parkinson's disease-like symptoms and give them lipoic acid, we've seen that there is huge improvement in the health of their dopaminergic neurons. And it definitely helps the neurons survive longer and do their function in a better way. We have seen that in our model. And for the mitochondria itself, we've done some mitochondrial measurements that I'm not showing here, indicating that lipoic acid also can help with the health of mitochondria in general. And there are other factors, which is a huge family of antioxidants like lipoic acid, and these help mitochondria work better. But in our case, we're showing a specific genetic pathway that's known to produce a natural product. And what we've done is we are able to put the gene back in the animals that lack it, the Parkinson's disease-like animals, and just putting the gene back is helping as much as putting the lipoic acid. So it's a linear pathway, we think.
Okay. I think that answered that. Next question is, do you anticipate that your gene therapy could be used in combination with other PD gene therapies? I think you kind of answered
I hope so. That would be nice. As we said, the disease is very complicated. It will be up to the doctors to see where it fits and, looking into patients and see what exactly causing the Parkinson's disease in a specific individual. And if this gene is one of the genes that were shown to be affected and we have the therapy at the time, it would be nice to have it too.
Well, as you said, that these will be kind of a library of therapies that would be for people who demonstrate Parkinson's disease, because not all one answer solutions are there for just the single disease.
Exactly. Yep.
So there is another question which I thought was kind of interesting. Some research has shown that Parkinson's is more common in males than in females. Could have the gender of the worms, if that is even measurable, played a role in how they were affected?
That's a really fascinating question because, believe it or not, the dopaminergic system or the dopamine neurons structure is a little bit different in males than females. So males have few extra dopaminergic neurons helping them with the mating process. I'm talking about the worm here. The female, or we call it the hermaphrodite because not actually female, has fewer number of dopaminergic neurons. And we are not sure if this is contributing to what we have seen now. We didn't do male versus female experiments yet, but that's something would be in our future directions to do. It's a very good future directions to go. I would like also to mention that one of my colleagues at the Brain Institute working with different gene, and she picked up so many differences between males and females in the sense. I mean, she more interested in ADHD and but also because it's associated with dopamine dysregulation, dopamine problems. We think it could be the same in or very similar in our scenario too.
I see. Another question came in from Gabrielle Felix. This was fascinating. Thank you for your lecture. I'm interested in knowing how this discovery can branch out to other neurodegenerative diseases. I'm particularly interested in CTE and tau therapies in general, and it's believed that those are correlated to dopamine depletion as well. Do you believe it would be possible to introduce tau proteins to C. elegans in a similar scenario, like it was done with the I'm going to probably butcher it.
A-synuclein, yeah. I think it's, if I'm not mistaken, there are models in C. elegans that have the tau protein for sure. And these were models for Alzheimer's disease. And actually, the antipsychotic drug I just talked about, azaprone, was used to treat these worms with the tau protein, mimicking Alzheimer's disease. So a lot of studies actually going in that other avenue as well.
I see.
And that's a very, very good question. Thank you.
And that is all of the questions. Now, I guess if there was any kind of final words, Dr. Refai, would you say would be the state of research in general for Parkinson's disease? Do you believe that we're making headway in the types of research that's going on, whether it be here at FAU or nationwide?
So at FAU, we have now a growing group of talented scientists who are tackling this problem from so many different angles, given the nature of the disease being very complicated. And we're hoping that with the support of our community and the great work of our leadership at FAU and our colleagues, we'll be able to move a step further and contribute to the national endeavors to solve this problem. I hope, and I always like to give hopes, positive hopes, not false hopes, that we are moving closer every day toward tackling this disease. We will be there one day, I'm pretty sure. It's still a few more steps to take, but we're moving into the right directions, looking into this from so many different angles. Researchers right here at FAU, at different institutions, and internationally, tackling the problem from different angles, given that the problem is so complicated. And putting these together one day would allow us to see the full picture and being able to describe a robust strategy to combat the disease.
That would be wonderful one day to see that happen.
Absolutely.
Well, I want to thank you for your time. And if there are any other questions, you're more than welcome to email research@fau.edu. We can get them to Dr. Refai.
Yes.
And with that, we're going to conclude the webinar. We really appreciate your time, Dr. Refai.
Thank you so much. It was my pleasure.
Thank you. Take care. Bye-bye.
Bye-bye.